Good afternoon, everyone. My name is Roshan Paudel, and today I'll be talking about exploration of taro breeding populations that show improved performance against diseases in Hawaii. So my talk will be focused on yield data as well as taro leaf blight disease resistance data of taro. So the taro breeding program has two objectives, broadly speaking. So the first thing we're looking at is agronomic traits such as disease resistance, and also looking at varieties that don't produce runners. And we also focus on quality aspects such as taste and color of the quorum. So this slide here shows the process of breeding taro, and I believe there is a publication that's coming out soon that will have more information, more detailed information, but I just would like to give you an idea of how to make a cross. So it's basically a simple process. The most important thing is to identify and select the flowers. Um, so the varieties that you want to make across. So once you identify the flowers, you have to look for mature flowers. So how do you know that? So basically a mature female flower will have will produce a sweet smell that can be detected from a long distance. In terms of the male flowers, mature male flower will produce pollen that has richer yellow or vibrant white color. So the next step is to cut the male part of the female flowers, and then collect the pollen and just drop the pollen off of the female flower, and then bag it to avoid any contaminations. And after 3 weeks, the plants will grow up to this size, and they can be transplanted into bigger size pots. So this is a general structure of breeding for resistance in taro. We have existing germplasm collection of local varieties, and then we also introduce taro leaf blight resistant germplasm from other sources, and we make crosses and develop breeding populations. And these populations are then put into a unreplicated field trials using a standard check cultivar because we are limited by um, the material in this stage. And then the selection is basically based on resistance to the disease, as well as we look at yield and taste of the variety. And then after selecting few cultivars, we then perform a statewide multi-year variety trials, and select the best variety and release them. So this is a conventional breeding method, um, but because we have molecular markers, we can also use them to speed up the breeding process by identifying genes and QTLs for the resistance, and then incorporating them into this breeding pipeline. So for today's presentation, I'll be focusing on the results of statewide multi-year variety trials. So this figure here shows the distribution of lines across trials. So in the y-axis, we have all these locations, 4 locations: Oahu, Molokai, Maui, and Hilo. And on this side, we have the different years that we did our trial. And these are all the varieties that we tested in our trials. So the color here shows the presence and absence of a particular variety in those environments. So if we have a pink color, then that means that the variety is not replicated in that trial. Now I want to talk about how we analyze our data. So we don't have all the varieties common in across all trials. So what we do is we use a common check cultivar, and then look at how it performed in particular environment and use that information to compare
against different environments. So that allows us um, for comparison across different varieties without actually replicating them in each environment. So this is the model of analysis. Um, so these are some of the traits that we are interested in. So we are looking at fresh weight of taro, percent rot, which is the percentage of rot quorum material. And then we also look at percent dry matter and taro leaf light rating. And this figure here shows the correlation between these different traits. And as expected, we see a strong correlation between fresh weight and dry weight here, shown by this dark red. And then also we can see that as the disease increases, the dry matter goes down, which is also expected. So this means that our data is pretty good. So as we conducted our trials in different years, we saw a similarity between certain years. So in 2016 and 2017, basically these 2 years behave in the same way as they are overlapping here. And then 2018 and 2019 behave in the same way. So there we saw a difference in environments across different years, which means that we are covering a broad range of different climatic conditions. In terms of fresh wet yield results, now this slide has a lot of pictures, but the most important thing is we saw certain varieties perform better than others. As you can see here, there's a bump in the yield performance, and this particular variety is 1016-03, and then recently we have named it as Ho'onui, and this is one of the varieties that is ready for release. And the other important um, result that we saw is these varieties, or particularly this variety, is performing consistently across different locations and different environments, and that's a good reason for selection of this variety. In terms of corn rot, we did not see much variation across years, but in certain locations there was higher rot compared to the other locations. So there was a location effect um, for the corn rot disease. The take-home message from this short presentation is we found some superior lines that perform consistently across different environments. And also we saw a good disease tolerance in these selected lines. But oftentimes while breeding taro, we see a problem of the resistance trait being linked with runners. So we have to carefully select varieties that do not produce runners. And also there is a environmental effect linked to this particular trait. So that brings to the end of my presentation. If you have any questions, please type them in the chat box. Our moderator for the Q&A session is Dr. Mike Shintaku. Thank you so much.